Welcome to The Explainer. Today, we're diving into one of the most incredible stories in all of biology, how we create gametes. We're going to follow the microscopic journey of the cells that become sperm and eggs, the literal building blocks of the next generation. So here's our game plan. We're going to trace this whole story right from the very beginning, starting with the mysterious origins of what are called primordial germ cells. Then we'll get to this crucial fork in the road, where these cells have to commit to either a male or female path. And finally, we'll follow both of those journeys to see how each cell completes its final training, getting ready for the big moment of fertilization. All right, let's dive in. Our story starts with a really unique group of cells, the ones that hold the entire blueprint for every future generation, the primordial germ cells, or PGCs for short. So, what in the world are PGCs? Well, you can think of them as the ultimate stem cells for reproduction. They're the common ancestors for every single sperm and egg. But what's really wild is where they come from. In humans, they first pop up around the third week of pregnancy, not even in the main body of the embryo, but way out in a structure called the yolk sac. From there, these PGCs go on an absolutely epic journey. This diagram shows them on the move in a mouse embryo, literally crawling from the yolk sac, through the developing gut, and deep into the core of the embryo. And they have a very specific destination, a place called the genital ridge, which they're going to colonize and basically build into the future gonads. Now, PGCs aren't just any cells, they have some truly unique superpowers. While they're on this long trek, they perform a critical genetic reset. They wipe clean most of the epigenetic marks, these little chemical tags on the DNA, from the previous generation. It's like starting with a completely clean slate. In female cells, they even wake up the X chromosome that was silenced, and all the while, they are multiplying like crazy. In mice, the population explodes from about 100 cells to over 25,000 just during this migration. It's nuts. Okay, so once the PGCs finally arrive at the genital ridge, they hit a massive fork in the road. This is the moment of truth, where the gonad itself gets its instructions to become either a testis or an ovary. And that decision sets the course for the entire embryo's development. You see, for a little while, around the 6th week in human development, this gonad is what we call bipotential. It's totally neutral. It has all the parts in potential to go either way, testis or ovary. It's basically just sitting there waiting for the signal. And that signal is controlled by this complex web of genes, kind of like a molecular switchboard. Now, I know this looks complicated, but the entire process really comes down to one master switch. In a genetically male embryo, the one with an X and a Y chromosome, a single gene on that Y chromosome, called SRY, flicks on. And that one little event triggers a huge chain reaction that builds a testes. This right here is the genetic domino effect for making a male. The SRY gene knocks over the first domino, which is another gene called SOX9. SOX9 then activates something called anti-Müllerian hormone, or AMH. And AMH has a very specific mission. It seeks out and dismantles the Müllerian duct, which is the structure that would have become the uterus and fallopian tubes. So it's an active process of building one pathway while simultaneously taking apart the other. So when you boil it all down, we have two very different strategies. Male development is an active process, kicked off by that SRY gene switch. But if that switch is never flipped, the gonad just proceeds down the female pathway. Now, this isn't just a passive thing, though. The female pathway is actively supported by its own set of key genes, like WNT4, which makes sure that the Müllerian duct sticks around and develops properly. All right, the testis is formed, the path is chosen, now the stage is set for spermatogenesis. And this is a truly remarkable process of non-stop sperm production that kicks in at puberty and can pretty much last a lifetime. The adult testis is basically a super efficient factory. It is packed with hundreds of meters of these tiny coiled tubes called seminiferous tubules. This is ground zero for sperm production. Once they're made, they get collected and moved over to the epididymis, which is this long tube where they'll go for their final training. And if we zoom right into a cross-section of one of these tubes, you can see it's a perfectly organized production line. The newest recruits, the stem cells called spermatogonia, are on the outer wall. As they divide and grow up, their kids get pushed further and further inward toward the center opening. And this whole operation is managed and supported by these big nurse cells called Sertoli cells. The entire production cycle, from start to finish for one sperm, takes about 65 days. And you can break it down into three main stages. First, you've got proliferation. This is a numbers game where the stem cells just multiply to create a huge workforce. Then comes meiosis, the really crucial step where the chromosome number gets cut in half. And the grand finale is differentiation. This is a radical physical makeover where a simple round cell gets sculpted into that sleek, streamlined shape of a mature sperm. One of the most insane parts of this whole makeover is the DNA packaging. Get this, a human cell has almost 2 meters of DNA inside it. To cram all of that into a teeny tiny sperm head, the cell does something incredible. It literally throws out its standard packaging proteins, called histones, and swaps them for much smaller ones called protamines. This lets the DNA get packed so tightly that it becomes completely shut down. A perfectly protected genetic payload, all ready for delivery. Okay, so now we switch gears completely and turn to the female pathway, oogenesis. And honestly, it could not be more different. Forget everything about a continuous factory. This is a story about a finite resource, one that's managed over decades. It's a process of long pauses and a carefully controlled dwindling supply. Now, a really key idea here is that the oocyte, the egg cell, never develops by itself. It's always protected inside this support structure made of other cells, and we call that a follicle. The oocyte and its follicle mature together, like a team. It's a partnership that starts way back in the fetal ovary and continues right up until ovulation, potentially decades later. And this brings us to what is maybe the single biggest difference between making male and female gametes, the ovarian reserve. While males are constantly making new sperm, females are born with their entire lifetime supply of oocytes already there. That's it. It's a finite, non-renewable resource. This graph really tells the whole story. It shows just how dramatic the decline is. The number of follicles actually peaks before a female is even born, at around 6 to 8 million. By the time of birth, that number has already plummeted to about 1 to 2 million. And from there, it's just a steady, irreversible decline until almost none are left at menopause. So you might be wondering, what happens to all those millions of follicles? Well, the vast majority are lost through a natural process of programmed cell death called atresia. In fact, more than 99% of all the follicles a female ever has will simply degenerate without ever getting the chance to ovulate. Each month, a small group of them is recruited to grow. But it's a ruthless competition where usually only one winner makes it to the finish line. The final maturation for that month's winner is triggered by a big surge of luteinizing hormone, or LH. This hormonal go signal kicks off a very precise, timed series of events. Meiosis, which has been on pause for decades, suddenly wakes up and resumes. The oocyte quickly finishes its first division, only to pause again at a stage called metaphase 2. And it's in this arrested state that it gets ovulated, about 38 hours after that LH surge first began. All right, we are in the home stretch. Let's look at the very last steps these cells take to become fully ready for action. Because for sperm, leaving the testis is just the start of their training. And for the oocyte, well, its final act is still waiting for one last critical signal. The epididymis is basically a boot camp for sperm. When they first leave the testes, they're rookies. They can't swim right, and they're totally incapable of fertilizing anything. But during their slow, 2-week journey through this long, coiled tube, they go through a total transformation. They learn how to swim straight, their membranes get modified so they can recognize an egg, and their whole metabolism gets fine-tuned. Only after they graduate from this boot camp are they ready for their mission. So, from that tiny cell migrating through an embryo to the final intricate dance of making a sperm or an egg, we've seen this absolutely mind-blowing biological choreography. Understanding this incredibly delicate and complex process is just so fundamental. It doesn't just show us the basis of how life continues, it also shines a massive spotlight on all the different points where this intricate process can go wrong, which has huge implications for fertility and the very beginnings of life itself.